Good evening, everyone, and a very warm welcome to today's webinar on long read sequencing of alternate isoforms for target discovery, brought to you by Strand Life Sciences. Leading today's session is Dr. Badri Paduka Sahasram, Vice President of Variant Science at Strand Life Sciences. In his career spanning for 18 years, Dr. Badri has worked in various capacities in companies like Roche, Illumina, Henry Ford Health Systems, and etc. Dr. Badri is a PhD in computational biology, population genomics from the University of Southern California and has done his BTech in biotechnology and biochemical engineering from IIT Kharagpur. I welcome you, Dr. Badri, and I please request you to please take the session forward.
Thank you very much, Kushbu. Hi, all. Welcome to the webinar. So today I'm going to talk about the utility of long read sequencing for uh, target discovery on the basis of uh, sequencing alternate isoforms. So I think our interest was to look into the this particular type of data, explore its utility for target discovery, and develop some initial pipelines that can do this type of analysis for us. And we applied that pipeline to an example cancer dataset, and that will be the primary focus of the talk. And then lastly, I will talk about some future improvements and refinements that are possible for this pipeline. Uh, so alternative splicing, as many of us know, is it's a fundamental post-transcriptional regulation mechanism that contributes to the diversity of the transcriptome and proteome. This type of events generates multiple types of mature mRNAs for a particular gene, referred to as isoforms, and nearly 90% of human genes undergo some type of alternate splicing. It has significant roles in many essential biological processes such as cellular development and differentiation, and aberrant splicing has been implicated in a range of different diseases like neurological cancers, metabolic and immune disorders. So, what is our goal here? So, our goal is to see if we can come up with uh, an algorithm or a pipeline that can use or leverage these alternatives isoforms and identify which ones may be important for a particular disease or a phenotype. And here, our main focus is on using or leveraging long read sequencing data for this purpose. And the main object of study in this talk is a particular colorectal cancer dataset to which our pipeline will get applied. Um, so. In At a very high level, so we have long read sequencing of say tumor and normal tissues, and then what we want to do is we want to see are any of these alternate isoforms. So first is how do we identify the new ones from the data? So a data-driven approach to identifying new isoforms. Then what we want to do is how to look for the ones that may be interesting for a particular disease. So which ones are specific to cancer and which ones might be potentially candidate targets for cancer. We want to shortlist or yeah, prioritize such types of isoforms. Then we can also prioritize by their putative functional roles based on prior knowledge of gene functions and pathways. Also, because our main or end goal is target discovery, so we are also focusing on what type of targets are potentially druggable. So either with small molecules or with biological agents, or such as antibodies or therapeutic proteins. So this is another type of filter that we want to apply eventually to get the most promising candidates for our purpose, which is what are some novel isoform targets, alternate isoform targets that can serve as targets for maybe like a pharma company to further investigate them experimentally. So to this end, we set up an initial pipeline on the basis of tools that can help us with this problem, and then we applied this pipeline prototype to an example dataset from colorectal cancer, and we discussed the results. I will discuss the results of that analysis, and lastly, I'll talk about future directions of how we can further refine and improve that pipeline. So overall, this flowchart gives us an overview of the pipeline we created for the purpose of identifying targets that are pertinent to a particular disease on the basis of long read data. The pipeline uses a combination of long read and short read information from individuals to boost the accuracy of detecting particular novel alternate isoforms. One of the main components is the use of both long read and short read data in our investigation. Um, we leverage the support from. One thing to note here is, so the long read data comes from Oxford Nanopore, and we all know that the ONT data is of relatively less quality compared to a short read sequencing like Illumina. So we know that short reads give uh, a much higher quality of reads, but at the same time they are limited in their length, and so they may not cover the full length of a transcript. Long reads, on the other hand, they could span the entire transcript, and so we can know the full combination of exons in a particular transcript. But I think these are limited by the quality of the sequencing data. So by leveraging long read data to identify and then short read data to do quality control, we can further strengthen and gain greater confidence in any new type of isoforms that we identify from our data. So we use a combination of these two methods to identify new isoforms, um, long read and short read. Then these undergo some type of annotation or classification into whether the isoforms are already existing. That means, do they match fully previously known isoforms in GenCode, the compiled list of known isoforms in GenCode, or do they represent something novel? Novel can have two types of classifications. One could be that it's a new combination of known splice sites. So that's one type of novel isoform. So the splice sites are all previously reported and known, but it we are seeing a new combination of them. Or you could have like genuinely new splice sites. So there are two types of novel isoforms that are possible in this type of data, and we try to look into all or both these types of isoforms, and we also look into some gene fusions that also come from the tool. uh, Once we have identified the isoforms, so I'll describe in a bit more detail about the procedures and the QC steps we undertake. But then once you have the isoforms, then the next question is which ones are likely relevant to your disease of interest. So here we apply this additional filtering. We are looking for recurrent isoforms that we repeatedly seen in say 10 or more cancer samples, but not seen in controls. That can be something that indicates that perhaps that's specific to uh, that particular disease and might be influencing the disease in some way. So that's the next filter. So once we have identified isoforms, we really want to check if it is specific to the disease. Then what we look at is the next step is, so we have an isoform, but how do you know that it generates a viable protein? To do this, we also do a translation of the open reading frame, and then translate into the protein sequence, identify open reading frames, translate it into the protein sequence, and then check for some criteria that might indicate that it is generating or possibly generating a viable and functional protein. So that's the next filtration step to check the protein sequence implied by the isoform, and then check for whether that particular protein is likely to be viable. And then subsequently, we also do some analysis of pathways. So once we have, so after the translation, and say we have applied some filters on the isoform and the protein implied by it, we want to say see if are there certain pathways that are overrepresented in this group of genes or proteins. So we did an overrepresentation pathway analysis to see if certain pathways emerge as significant, much more than chance. So that analysis was carried out, and then specifically we used the wiki pathways, wiki cancer pathways database for this purpose. And then we also did an intersection with the druggable genome, because we are interested in uh, targets that might translate that has the best chance of translating into drug discoveries. So we intersected our candidate list with the small molecule and the biodruggable genome, and that gave us a final list of candidates that can potentially be followed up in order to see if new therapeutics can be designed for those targets. So that's an overview of the pipeline, and I think the key step is this use of long read and short read data. So long read to identify the new isoforms, and short read support to do the quality control on the new isoforms. Um, these are some of the steps and tools that go into the pipeline. So we align the long reads with the splice mode, then look for the splice junctions and the isoforms implied by that alignment. Then we also use an aligner for short reads. Then I think one step that's applied is called a correction step. So the correction step, what it does is it takes our splice site that is identified in our particular sample, and then it compares against known splice sites in the GenCode database. So if a particular splice site we see is within a certain window of known splice site, and then it corrects and fixes it to the known splice site. So here the interpretation is that if you are within a certain distance of the known splice site, then it's likely like sequencing or alignment artifact, and so those get corrected. But if you are further away, if it's a previously unseen splice site, if you're further away from known GenCode sites, and then you also have short read support for your new splice site, then those are of course retained. And then with this type of filtration steps, then all the transcripts with the similar or the same pattern of splice sites, those get collapsed, and ultimately we get unique transcripts from our tool. So the tool is referred to as FLAIR, Flare tool. And then lastly, any retain, so any transcripts that survives all our filtration steps, these are then getting annotated into few different categories, which I will describe in the subsequent slides. This figure is giving an overview of the different steps in the FLIRE methodology to identify identify new and new alternate isoforms, as well as existing ones. So we do first, like as I said, some correction of splice junction depending on how close it is to the known gen code site. So the choice of this of course can be varied. So the default value is like 10 base pairs. So if in the flare tool, so if a site you see in the long read data is within 10 base pairs of a known splice site in GenCode, then that transcript is retained. Of course, we are also filtering by the support for particular new new splice junctions. So in the previous step, so if it is within that k or 10 base pair window, then that undergoes correction. It gets corrected to the known gen code site. If a new splice site is further away from more than 10 base pairs from a nearby gen code site, then we check for support. So is it supported by multiple long reads, and is it also supported by short reads? If there is sufficient support, then that gets retained, and we can be more confident of such an isoform. And then yeah, if there are multiple isoforms of same pattern, these are collapsed, and ultimately FLAIR gives us like a set of unique isoforms we see in the sample. And subsequently this we can also do based on the counts some type of quantification. We don't do that in this analysis, but it can potentially a future task which we are planning using the expression level quantification of the specific isoforms. Once we have identified isoforms, then what we can do is we can uh, try to categorize them into a few different categories. So an isoform that like fully matches a known gen code transcript is referred to as a full splice match, and sometimes it can be an incomplete splice match. The two main categories that we are mainly interested in is the isoforms that are novel, not previously seen in the gen code. This can be categorized into two different types. One is novel but in catalog, which means you have a novel combination of splice sites, but all the individual splice sites are in the database. But you have not seen that combination of splice sites before. Then you could have an entirely new splice site, which is this other group called the novel not in catalog um, category of isoforms. So generally, we focus mainly on these three groups: FSM, NIC, and NNC, which account for the vast majority of our transcripts. There are other rare ones, but we haven't looked more into that. We want to look into novel isoforms. Then we want to see if Some of them can be linked to diseases based on their relative presence in disease versus controls. Um, so that's the gist of our pipeline that we created for identifying putative targets from sequencing of alternate isoforms. And then we applied it to a public data set from colorectal cancer study. So we have 50 colorectal cancer data and 10 healthy controls, and we have both long read and short read data here, and the coverage and the read lengths are mentioned here. Um, so so we started out with our initial discovery of isoforms from our pipeline. We used long read and short read information, um, and then we applied the alignment, then the short read alignment, then the FLAIR tool to that uses both short read and long read, as well as prior gen code annotations to correct and collapse the isoforms, and finally gave us a list of candidates. Of course, the initial set of candidates is quite large, but then, as I mentioned, there is a because we are interested in isoforms that may be relevant to a particular disease, and then there is some other additional criteria like generating a possible functional or viable protein, being druggable by small molecules or by biotherapeutics. As we applied these additional filters, we found a reduction in the number of candidates, and we get a greatly reduced set of candidates after the application of filters. Of course, we can as we work more on this, we also have other ideas that can further narrow down or help us further prioritize among these what are the most promising or viable candidates, so which I will describe towards the end of the talk. But as you can see, so just applying, looking for things that are specific to disease, that greatly reduces from a very large candidate set to like few thousands. Then uh, for something to be likely to generate like a coding protein, that we did these few criteria. So first is we said that the ORF uh, should be greater than or equal to 300 nucleotides, and so amino acids should be greater than or equals to 100. And then also for the isoforms, so we first translated that into the protein sequence, and then we did a blast against the UniProt database and only retain the ones that have certain level of identity with known proteins. So with these two basic filters, we can further reduce our candidate set to a set that's likely to produce a viable protein. Note that there could be more sophisticated computational way to do this filtering, and I will talk a little bit about that um, in the future directions part. But as we apply these various filters, we get a prioritized set of candidates, and basis of these candidates, we also carried out a pathway overrepresentation analysis on the basis of wiki pathways cancer database. um, and this is a plot from uh, the results of the pathway analysis. The x-axis is the enrichment ratio, the y-axis is the negative logarithm of the false discovery rate, and I think among the different pathways, the The top two pathways that emerged include things like metabolic reprogramming in colon cancer pathway and then cell cycle pathway. So these emerged as the top hits, and they are highly statistically significant, as you can see from the FDR levels. Then we looked into which of our genes from our previous list intersect with these particular pathways, and then we wanted to see if there are particular candidates that emerged as promising targets. With the metabolic reprogramming pathway, about like we have 19 genes that are listed here that survived all our filters, and that are part of the metabolic reprogramming pathway. And we have also shown like the how many isoforms we are seeing, and whether they are druggable either by small molecule or biodruggable. So among these 19, several of them are druggable, and might be putative targets that can be pursued further. Similarly, for cell cycle pathway also, we have 31 genes, and we have indicated the druggability status, and there are several like HDAC1, CHEK2, and CDC25B that are druggable, and we can potentially pursue them or do further downstream analysis on them to look into which of these can be pursued further. This is overall an example of how we can identify alternate isoforms, then look for isoforms specific to disease, and then apply a range of filters to see which ones could be potentially new targets that could be pursued. Um, so that's overall what we did in this analysis. Then I am here showing some examples of IGV view from the IGV viewer, some examples of new alternate isoforms. The last row, the ref select in blue, so that is the canonical transcript for the GA PDH gene, which is one of the genes in the metabolic reprogramming pathways. And then you can see that this has like many smaller exons here. On the other hand, we have a new transcript here, which has like four different exons, five different exons, and not all the splice sites in the canonical transcripts are seen in this new transcript. So it's an example of the type where you are seeing a new combination of existing splice sites. So here the first row is the long read data, the first block of results. So this is then new isoform identified by the long read data. Then the second block is the results from the short read data, and we can see that these splice junctions have support from the short read data, and then a combination of these two, that we have both long read and short read support, gave us greater confidence that this is less likely to be a technical artifact. So we have much fewer exons than the canonical transcript in this new identified isoform. Similarly, another example from a cell cycle pathway gene, ANAPC5. So here we can see that there is only one exon in the blue here towards the right hand side of the figure in the canonical transcript. On the other hand, we are seeing in our long read a new splice site. So this is an example of a novel splice site not previously seen in the annotated data, and also there is short read support for that new splice site. Yeah, looking at both this evidence can strengthen our confidence in particular new isoforms that we identify. Help us improve the reduce the false positive rates in our discovery step for the new isoforms. That's mainly our results. We have developed a comprehensive pipeline that can shortlist alternate isoforms using a combination of long read and short read data. We have applied it and demonstrated it on a particular publicly available colorectal cancer dataset. Um, so the short read helps with quality control. Then once we have made discovery of a certain number of isoforms, we do apply further filtration steps, and this is based on the translation of the isoform into proteins and looking at the implied protein sequence and how viable that protein is, and what is the predicted impact arising from that protein. So that type of filtration helps us further narrow down the set of candidates, and also we look for things that differ in their relative frequency between disease versus control, and that also helps to narrow down further the set of candidates. Then additional filters included like the druggability of that particular target, which further narrows down the number of the number of possible genes. Also, we can look into the pathways and the predicted gene function of this list of targets, to see whether to ascertain whether they are biologically plausible, if the pathways or the known function of that gene can be related to cancer. We can do all these downstream steps after that initial discovery of possible cancer-specific new isoforms. A range of future work can be carried out to further strengthen our downstream analysis. This could include things like looking at the cellular localization of the protein of interest, looking at tools that can. So all this is focused on the at the protein level. So let's say we have an isoform, we have a protein sequence predicted by that isoform, and then we can look for the functional domains and look at the predicted structure of that protein. So this can also help us further choose among our candidates to see which ones are the most promising ones for the target discovery, and pursuing drug candidates. Uh, so I indicate a few different directions for future work. So this was the initial pipeline we created and showcased on this colorectal cancer data. But I think several improvements and refinements are possible. First is we have used the FLAIR tool for the new isoform discovery, and we can also explore the utility of alternate tools. There are also other newer tools like isoquant, and which have shown better performance than FLAR in recent benchmarks. So that's one exploratory analysis. So we can compare a range of different tools at this isoform discovery step. Then coming to the step of identifying disease specific isoforms. So what we have applied right now is a basic criteria. Essentially, we had this stringent criteria that we repeatedly see like 10 times or more the new transcript in the cancers, but never see that in the control. But here we haven't considered the abundance of a novel isoform. in our analysis. So we can do follow this up with more a differential expression analysis with tools like Salmon or Callisto or other relevant tools, and we can check for some relevant metrics or that for which we can look for differences among cancers versus controls. That's one line of investigation. Then, with regards to the testing the viability of a particular isoform, also we applied a relatively basic filter. We looked for isoforms that generate a protein that's 100 amino acids or longer, then checked for identity with known proteins in the UniProt database. But we can also carry out uh, more sophisticated computational or machine learning approaches to predict whether or not a particular sequence in the isoform will generate a coding protein or not. I will briefly talk about it. And then, last but not the least, we want to explore the impact of that protein implied by the isoform in much greater depth. So here we are interested in use of AI models such as AlphaFold and AlphaFold Multimer or ESM fold that can predict the sequence from the sequence, predict the structure of a protein. So this can be carried out for the novel isoforms, and then we can look into what that predicted structure implies in terms of whether certain interactions or certain binding sites or active sites are getting disrupted as a result of this new isoform. So for the differential analysis, in addition to like looking for things that are exclusive to cancer, we can also look for things that show a great difference in the abundance between the cancer versus control. So we can either look at the absolute abundance differences for a particular isoform, but we can also look into this quantity called isoform usage. So isoform usage is what proportion of the transcript of a gene correspond to a particular isoform. So this the what proportion of the transcripts, the frequency, the relative frequency of that isoform compared to the other or the overall total number of isoforms. So this can also be indicative of something related to cancer. So for example, if the frequency of a particular isoform greatly increases in cancer, might hint us that maybe that the usage of that particular isoform might be driving some pathways for that disease. So we can carry out a differential analysis. So in addition to just looking for things exclusive to cancer, then as for checking the viability of a new isoform, there are machine learning tools such as potential calculator or coding potential assessment tool that can give you a probability between 0 and 1 that a uh, particular isoform is likely to or can generate a coding protein. So this type of tools can also be leveraged to prioritize among our list of candidates, maybe which are the top isoforms we want to pursue. So this type of probabilities can also be useful. And last but not least, we want to look into, so once we have a further narrowed down list, we also want to look into uh, yeah, what does the isoform imply in terms of a protein structure, and what does it how does it impact, say for example, if the canonical protein has interaction partners, what does that imply for those interactions? So there are tools, AI tools, I think AlphaFold from Google and AlphaFold Multimer also from Google, and maybe ESM Fold from Meta that can help us answer this question. And we can check for disruption of functional sites as a result of an alternative isoform. So, for example, if if the isoform disrupts the interactions between the partners of the original protein that might indicate maybe a functional impact or a damaging impact on the relevant pathways. So these types of things we can check once we have the protein sequence. We can check if it removes a post-translational modification site. So all this type of further exploration at a protein level can help us further filter down and get the most biologically plausible or most promising candidate for target discovery. Yeah, thank you. That's it from me.
Thank you, Dr. Badri. That was a wonderful presentation. Now, I would like to request the audience to please raise your hands or post in your queries if you have any. Yeah, Mr. Santosh, you can please unmute.
Yes. So, good evening, all. Dr. Badri, thank you for nice presentation and very knowledgeable information. So, I was just trying to understand, you started the talk with the you are working in the colorectal cancer and the very high depth of work you in future direction you mentioned that like isoform expression and isoform variant the way like mechanism what you are going to use AI and ML tool how like these are this could affect the I mean the post translational modification maybe binding site or something. So the my simple question is sir, uh, is there any disease have been any cancer like have been studied by this isoform like this combination of long and short reads? Are they publicly means like it is in use because some I guess some validation is needed at this level.
Yes, yes. I think so. This is a public data set, so it has been studied before. So we have, I think, yeah. So definitely, cancers have been studied with for looking for alternate isoforms. I think the validation part, the experimental validation that I have to check for. But I think the most of this is with orthogonal validation from the predicted protein structure and so on. But I think what we want to do ideally is eventually, so let's say I get an isoform, I want to look at some proteomics data and first actually see that that particular isoform is supported by protein data. So we can look at some mass spectrometry data from colon cancer, and we can check or other tools,
Yes, yes,
maybe
yes,
some more. I think ideally, I mean, even better would be if we can study the entire protein rather than, because mass spectrometry, the problem is it only gives you chunks of peptides.
yes,
So
yes,
we
yes,
may not fully be confident still whether that entire isoform is seen, but definitely any tool that can give us whole maybe larger protein sequences. So ideally that type of data we have to look to validate these any promising. So first we have to establish that that particular isoform is actually expressed and we actually see evidence for that altered protein. That would be the first step, and then
yes,
subsequent follow-up steps should
yes.
happen later.
Yes. So I thank you for your nice explanation. The idea looks very promising, like finding isoform, their expression, and the protein level. So my simple question, and I totally agree with you that the mass spec analysis. So what is happening at the level of protein? So what are the protein being AI tool, ML tool definitely help in narrowing down the targets or something but validation. So I believe, and you mentioned, sir, one point of time some druggable like the pathway analysis or something like that. So how we can link this thing like pathway? You mean to say like the metabolism, like how drug metabolize, like something like that, or means how it is going to help in the drug level?
Uh, yeah, yeah. So I think first is so once we have a target, so not every target can potentially lead to drug, because like if it has to be like a small molecule drug, so that that particular protein has to have certain pockets which the molecule can bind to and inhibit. So there are so depending on the structure of the protein, people have created these lists of druggable targets. So not every target can have equal value
I
for.
agree. I agree. I
That's
agree.
one
Yes.
point.
Yes.
And then the other thing is, yes, I think if we have a like a uh, if we get a new target, then we also want to establish some level of biological plausibility. So if we can, if we have, I mean, if we have some prior knowledge of that gene, that we can come up with a hypothesis that you can, if it is involved in a certain pathway and that pathway has been implicated in cancer, then we have a plausible hypothesis of a mechanism for how that target might work. And that's why we need understanding of the target. So any new targets, we look into what pathways it's associated in, so that we can come up with some hypothesis for its mechanism of action. So right now we only know that it's associated with that disease, and it's more frequently or exclusively seen in cancer versus control. But to give us more confident, we also want to have some putative hypothesis for the mechanism of action. So that's where this pathway analysis will help. Then the druggability will help, like from a practical point of view. If a pharma company wants to pursue, it will probably pursue
Yes.
first the druggable
Correct.
targets. Yeah.
Sir, one question just based on your experience, I would like to understand. You told that like you are using nano for making long reads thing, and then you are correcting with the short reads and finding consensus. So since long reads have improved library wise, quality wise, and all lot. So do you think that based on your experience, like the short reads improved how much in terms of percent, uh, like if it is, can if someone is interested in pursuing only long read and making their study, is it good idea or both short and long are needed?
Uh,
I mean, how much differences you observe after mapping and all these things? That is my question actually.
Yeah, that's a good question. I have to look into that numbers. But I think so generally, yeah, we expect the precision to improve with the short reads because we are reducing the false positive rate. But we haven't precisely benchmarked one versus the other. I will try to look for literature. I will share it with you. But there have been some benchmarking studies that have been done. I will try to point that to you, but we didn't look
Okay,
that
okay, okay,
investigation. So
okay, sir. Thank
yeah,
you, sir.
look into that.
So thank you for answering my all queries. Thank you, sir. So it was very nice presentation and good learning. Okay, sir. Thank you.
Yeah, thank you. Sure.
Am I audible?
Yes.
Um, I'm having some glitch. I'm not able to hear, but
Oh, are you able to hear?
one.
Yeah, yeah, it's audible. Yeah.
Okay.
Uh, Anyone else in the audience, if you have any questions, please go ahead.
Yeah, sure. I think they are just asking for the meeting recording. Yeah, we will share it.
Are there any more questions? If not, thank you, Dr. Badri. And if anyone has any more questions, please mail them to us and we'll get back to you.
Okay. Thank you. Thank you all for
Thank you so
attending.
much. Yeah.
Thank you. Thank you.